final exams. How do you want the recording to, I mean, the exam to be MCQ based or anything else? I think MCQs will be
So
fine.
MCQ. Yes, sir.
Yes, sir.
Yeah,
So we will
MCQ
also
will
get
be
an idea for the gate
Yes,
as
sir,
well.
MCQ.
great. MCQ. We'll have two kind of questions, multiple choice and multiple select questions. There'll be obviously no negative markings, but yeah, it will be a good way of assessing how things go on. But anyway, we'll discuss about the assignment in the end. Today we'll be discussing mostly about nucleus. So these three particular hallmarks, genomic instability, telomere attrition, and epigenetic modifications, they are all associated with the nucleus. So nucleus plays a very important role. You know what a nucleus is, right? All of you know that.
Yes, sir.
So nucleus is a separate compartment in eukaryotes, which is used to store chromosomes. So it's a highly organized repository of an individual's genetic information. It contains chromosomes and proteins. These proteins have some functional and structural roles. So we'll talk about some nuclear architecture, mutations in DNA and DNA damage repair mechanisms, all which happens in the nucleus. So this is how the nuclear membrane looks like. So there is the the outer membrane and the perinuclear space in between. Then there is inner membrane, and below that there is the nuclear lamina. This is the nuclear lamina, which is supported by proteins called lamins. We have talked about lamins, if you remember. Do any of you remember about lamins?
Yes, sir.
Okay, so we'll again talk about it, and below that there is chromatin, and there are these nuclear pore complexes. These complexes are important to take out any material or take in any material that is required in or out of the nucleus. So that is important. Then we have the rough endoplasmic reticulum, all these kind of cell biological aspects. We have the ribosome. Why is the ribosome important? Anybody?
Sir, ribosome is the basic protein factory of a cell.
That is correct. So DNA is there inside the nucleus, then the mRNA is produced, mRNA is taken out, and then the ribosome, it translates into the respective proteins that we are talking about. Anyway, that is what happens. So if you see the size and shape of nucleus of a nuclear membrane in this case, so these are how C elegans look like. This is the structure of a neutrophil. And this is what we observe in aging. So what do we observe? What can you observe here? This is the nuclear membrane we are talking about that has been stained. So any particular observation in that end?
Sir, what do these pictures symbolize?
This is these are the cells taken from a 9-year-old person. This is from a 96-year-old person, as mentioned. So what happens when you age?
The cells atrophy.
Yeah, there is a change in the structure. It forms a blebbed structure. Something like that happens. So we'll talk about how that happens. So we talked about lamina. Lamina is a network, inside the nuclear membrane. Have you do you I mean, have you guys studied about these proteins or these structures before?
Yes, sir.
About lamins and
Yes,
nucleus
sir.
nuclear structure? You have studied about it. Okay,
Yes.
good enough. So this will be a revision in that sense. So so what are lamins or laminas? These are the interwoven networks situated at the periphery of the nucleus, which underlying the nuclear membrane. So lamins, they form majorly the major component of lamina, and it forms a protein network adjacent to the nuclear membrane. Now it helps in maintaining the mechanical properties, the shape, and all these kind of things. Shape is maintained, the shape of the nucleus is maintained using these lamin proteins. They form a fibrillated structure which gives the shape of the nucleus. So this is where essentially all the heterochromatin, are also situated. So we'll look at it. We'll look at it one by one. So essentially, lamins, they are made up of three kind of proteins: lamin A, lamin C, and lamin B. Again, lamin B are of three types: lamin B1, B2, and B3. So lamin A is formed from element A gene, whereas lamin C is again formed from element A gene, but it is a splice variant. So you know what is a splice variant? All of you know what splice variant is? Anybody who doesn't know? Am I audible?
Sir, I don't know, sir.
Okay. So let's say you know the concept of exons and introns, right?
Yes, sir.
So let's say these are the exons E1, E2, E3. So these are the exons. So these exons will then together they will form the mRNA
Okay,
like
yes, sir.
this. That you know, right? So
Yes.
introns will be spliced off. So you know the mechanism of splicing, right?
Yes, sir.
So sometimes the splice variants are something like this. So let's say we have E4 here, something
So
like that.
Sir, are you drawing something or something?
Yes,
Right.
yes.
So that is not visible.
It is not visible.
No, sir.
Okay. Let me try again. Should be visible usually.
So it is visible, sir.
Huh?
It is visible. I guess it's some network problem.
Okay. I don't know how to solve that. Is it a network
Anyway,
problem
sir.
from
No, sir.
my end? Yes, Pragya.
Actually, sir, I have noticed that actually throughout the rest of the classes also I saw you writing or scribbling something, but it was not visible in my. Maybe because I have not joined with the college account or something.
I think others have also not joined using the college account. Uh,
Anyway, sir, continue, sir, continue.
okay, I'll do something. If it is not visible, it doesn't make any sense. I'll try to share the slides in a different way. Let's see. In this way, I cannot see you people, but anyway, you can see everything basically. Let's see. Yeah, is the slide visible now?
Yes, sir.
Yes.
Is it moving?
Yes, sir.
Yes, sir. Yes, sir.
Okay, so as I said, let's say, can you see it now?
Yes, sir. Yes, sir.
So these are the exons E1, E2, let's say E3 and E4. So these are the exons. These exons together will form the mRNA, right? Something like this.
Yes, sir.
E4. So sometimes what happens, there is differential splicing. So what could happen? Instead of this intron getting splicing, these two introns, they get spliced, even E3 gets spliced. So the mRNA formed or the protein formed finally contains E1, E2, and E4, right? That
Yes.
could be one variant. Then there could be another variant where, let's say E2 is spliced. Okay, this could be one variant, another variant where E2 is spliced. So where there we will have E1, E3, E4, something like that. This kind of protein we will get. So these two are from the same gene, but they are called splice variants.
Okay, sir.
Understood?
Yes, sir. Yes, sir. Thank you.
Okay, good enough. So lamin A and lamin C, these are splice variants coming from the same gene, while lamin B and we'll not be talking about lamin B as much. But anyway, B type lamins, they are expressed in all types of cell, every cell, whereas expression of lamin A is always is developmentally regulated, means with development we have this lamin A expression. So just a minute. Yeah,
Sir,
so
here the splice variants are the A and C, right?
these two. Yeah, A and C are splice variants.
A and C. Okay, sir.
So this is how we talked about initially about lamin formation, lamin A formation. So this is the structure of lamin A, how it looks like. So it has pre-lamin. Initially, it is pre-lamin, which has nuclear localization signal. Why is it important? A nuclear localization signal is important because it will tell that the protein needs to go to the nucleus. So what happens? There is a CAX motif where there is farnesylation. This is the farnesylated motif, which happens at the cysteine residue. Cysteine residue of the CAX motif, it becomes farnesylated. Farnesylation is a post-translational modification, right? Now, once it is farnesylated, what happens? There is a proteolytic cleavage. So proteolytic cleavage followed by methylation of this, moiety. So once this is methylated and cleaved, we get a mature lamin. So this is an important step in formation of this lamins, and these lamins are essentially they form the structure of the lamina, the nuclear lamina. This is what happens normally. But in case of Hutchinson-Gilford progeria syndrome, there is a mutation in lamin A gene which causes a genetic disorder and causes premature aging. So if you have seen the movie Pa, if you remember Amitabh Bachchan's movie, there we had this kind of a problem. So there is activation of a cryptic splice site and generates an aberrant form of lamin A, which we called progerin, which comes from the disease itself. So there is essentially 150 amino acid deletion. It happens in case of progerin. So it is a different form of lamin which is produced, lamin A essentially. So in case of normal healthy tissues, you can see this is a round structure of the nuclear envelope or the nuclear membrane, right? In the top.
Yes, sir.
So in case of progeria, what we see that there is a bleb structure, the nuclear architecture is, disrupted. So that is what happens. Now, vast majority of Hutchinson-Gilford progeria syndrome, it happens due to a single base substitution, that is GGC to GGT. Now, this substitution does not cause any change in the amino acid, but it causes a deletion of 150 nucleotides in exon 11. And therefore, there is an alternatively spliced truncated variant of lamin A mRNA, and there is an in-frame deletion of 50 amino acids. So yeah, I somewhere wrote 150 amino acid. It will be 50 amino acid, 150 nucleotide. So you know the math, how it is happening, right?
Yes, sir.
So one amino acid is coded by three
Three
nucleotides,
base
right? Yeah.
nucleotides. That's right.
So near the carboxy terminal, there is a deletion of 50 amino acids that leads to the change in the nuclear architecture. Now, this is how it essentially looks like. This is a normal, variant where there is a normal splicing. So exon 11 and 12 will be normally present. And then there are these, uh, CSIM motif, where again cysteine gets farnesylated and then it becomes methylated and then it is cleaved using ZMPTSE, which is zinc metalloprotease. It's a protease essentially. So what happens in HGPS protein? This exon 11, it has 150 nucleotide deletion. Due to this 150 nucleotide deletion, what happens? The protease site is also deleted, because of which this farnesylation happens, but the proteolytic cleavage does not happen here. Thereby, the farnesylated motif remains in the mature lamin also, or the alternative form of lamin. The farnesylation, it remains, and this farnesylated lamin is called progerin, which is toxic to the cell. We'll see what happens when this is present. So everybody understood so far? Is it
Yes,
Yes.
sir.
clear? Okay.
Yes, sir.
So
Sir,
this
that
is how.
sir, 1 minute. That farnesylation, previous slide.
Yeah, wait a minute. Yeah.
Sir, here here farnesylation is basically done so as to add that extra peptide there or to excise the peptide.
This farnesylation, or here also, it's an important post-translational modification. So it happens in our body. For what reason? Few people say it is for nuclear localization. Also, it is important. But once it gets into the nucleus, once it's its role is finished, at that point it is then cleaved off.
Okay,
So
sir.
we'll see. We people have tried to introduce some farnesyl transferase inhibitors also to treat this kind of diseases, and it has worked out. So we'll see what happens. So this is how a nuclear structure is. So if you see these are these nuclear membranes. This is the outer membrane. This is the inner membrane, and then there is the peripheral space. And below that, these green colored structures, these are the lamins, right? These are the lamins. These are the nuclear lamina. And one thing that you can observe is that all the closed chromatin structures, which are called heterochromatin, also they are towards the lamin, right? Can you observe that? They are all attached to the lamin.
Yes, sir.
This is also very important. So this has also been observed in case of nucleus. In case of progeria, what happens? You can see these red color structures. These are progerin. Now, this progerin can no longer maintain the structural integrity of the membrane. That's why the earlier what we had observed, that there were heterochromatin structures. Now we see that these heterochromatins have turned into euchromatins because the structure is no longer maintained. So is that understood to everybody? So if
Yes,
that
sir.
happens, if progerin gets accumulated, there will be an abrupt gene expression, right? Does that make any sense to all of you?
Yes, sir.
Good enough. Then let's go to the next slide. Here we see that again, H3K27, sorry, H3K27 methylation is a marker. It's a marker of heterochromatin, which is facultative. So you know what is facultative and constitutive?
Yes, sir.
You have studied about aerobic respiration. Must have studied about facultative
Yes.
anaerobes and all that kind of things, and constitutive anaerobic respirations. Facultative means when it is required, then there will be gene expression. When it is not required, then it will go back to the heterochromatin state. So these H3K27 methylation state, this is reduced in case of the HGPS patients that we have observed from the Western blot, as you can see here in the B part, right? Also, if you see the control there, you will see that lamin A, the structure of lamin A is changed. Clearly from this B panel, you can observe that that H3K27 methylation is decreased. This is just an overlay, which is basically combining both the images. So okay, the same thing has been observed in all these panels. In the F panel, what we see is EZH2. It's a methyl transferase. You know what is a methyl transferase?
Yes, sir.
Methyl transferases, they methylate proteins. So methyl transferase levels are decreased in HGPS patient. Makes sense because methylation is also decreased. So why are we studying this disease in particular in context of aging? Because in case of HGPS, we observe accelerated aging. Okay, these patients, they hardly live up to their teenage years. They die very soon. They have accelerated aging, and that's why it is a very good model to study aging. So if we actually we are able to save these patients somehow, those kind of drugs which will be used to save these kind of patients can also be used for aging, for anti-aging essentially. So is that clear to everybody?
Yes, sir.
Okay, so the same thing we see now. We see another methylation. This is H4K20 methylation. So again, H3 we observed H3, sorry, K27, and this is H4K40. So what is H in both the
Histone.
cases?
Histone.
Yes, histone 3 and histone 4. Good enough.
Histone
What is
4.
K
K is lysine.
and
27th
20?
amino acid and 40th amino acid.
Okay, everybody understands that, I hope. So that is fine. So H4K40, this is marker for constitutive heterochromatin. So something that we observe that constitutive heterochromatins, they increase. I mean, the marker for constitutive heterochromatin, they increase in HGPS patients and they decrease in normal patients. That is what we observe.
Sir, methylation, in this case, what does methylation do? Like in AGPS, the methylation rate of this H4K20, this increases. So it causes the heterochromatin to express.
Yes, right. That is correct. So when there is facultative heterochromatin, whereas
Yes.
there are some genes which needs to be expressed at some point of time, then it needs to be stopped. Then again, maybe at some time when you require that gene, you will again express it. So that kind of genes are regulated by
then what
facultative.
do we that means constitutive heterochromatin is not does not usually occur, but in the case of this extra methylation, constitutive heterochromatin is seen.
Constitutive heterochromatin is observed. It is definitely observed. I will tell you how. Let's say, if you have, some cell types, they only behave like their own cell types. Let's say if we take neural cells, they will only behave like neural cells. So all the other, genes associated with any other cell type, they will be completely switched off, isn't it?
Yes, sir.
So there we will require a constitutive, down regulation of certain genes. So there we will require constitutive heterochromatin. But anyway, in case of HGPS patients, we observe an increase in this constitutive, H4K40 methylation. Clear on that? We have seen this on HeLa cells. So HeLa cells, these are cell lines. One of the first described cell lines. So there we observe these, changes. Any doubt on that? I hope not. So as I said, the HGPS, their patient's life expectancy barely reaches teenage years, and they usually die due to cardiac problems like myocardial failure or stroke, or any kind of symptoms that are associated with aged people, like subcutaneous fat loss, bone and joint abnormalities, and so on. And they appear in a few months after births. Okay. So they age very faster, and the severity of HGPS, it might depend on the ratio between progerin and prelamin accumulation. So the more the number of progerin, the faster you will age, of course. So that's why progerin is toxic in that sense. And now we were talking about ZMPTSE. What is it?
Which actually cuts the farnesylated moiety from the prelamin and makes it functional lamin.
So it is a protease. Which kind of protease?
It's a exopeptidase.
Zinc metalloprotease.
As a zinc metal.
So where should it be present in the cell?
Inside the nucleus.
That is correct. But what we observe that this protein has dual localization. It is present obviously inside the nucleus, as you said, and also in. Wait, just a minute. Just give me a moment. I think, did I miss something here? Let me have a look. Okay, it is also present in the endoplasmic reticulum, ER. These two places. So the possible treatment strategies could be to prevent the downstream effects of progerin. These are some of the ways we can do that. So like JH4, as I said, JH4, it prevents the interaction between lamin A and the progerin. So there are different ways. Then there could be progerin clearance mechanisms where these kind of progerins, if they are produced, they are removed from the system. Then the toxicity of isoprenylated or methylated prelamins. So isoprenylation is essentially farnesylation. There are other kind of isoprenylations also, like geranylation, We'll talk about it again. So these FTIs, they prevent that things to happen. So there are some mechanisms to do that. We can prevent the aberrant splicing also, and potentially, probably we can correct these kind of diseases right where the mutation happens. So probably using CRISPR, but we did not have that much of advancement yet. So let's see. Um, so these FTIs, these are farnesyl transferase inhibitors. These are small molecules which reversibly bind to the farnesyl transferase CAX binding site. Now, blocking the farnesylation of progerin with FTIs, it restored normal nuclear architecture and resulted in significant reduction in nuclear blebbing, both in transiently transfected HeLa, HEK293 means all kind of cell lines essentially. Uh, then we have a small molecule drug, lonafarnib. It's a protein farnesyl transferase inhibitor. It has worked for these patients. And does the localization of lamin A alter? That was a question which people asked, because why is farnesylation important otherwise? So by introducing a stop codon at the end of lamin A coding sequence, one of the papers they discussed, they generated a lamin A knock in mice, where lamin A is synthesized directly, completely bypassing the farnesylation step. And then they still observed that still the the non-farnesylated lamins, they were still going to the nucleus. So the importance was not clear, at least in case of mice, because they studied in mice. So so are the non-farnesylated lamins innocuous, or they may promote disease? So what is their importance that we should ask? So they tried to change the cysteine residue. So cysteine residue, which actually gets farnesylated, to serine residue. So that will prevent farnesylation. So they observed that the disease had a milder phenotype. There was less expression of progerin, but again, we are not sure that why that milder phenotype is coming up. Then again, this CAX motive, it was again then changed to CSIM to CSM. There we saw that CSM progerin was non-toxic. So these are some studies which was done to trying to understand what happens. Then alternate prenylation, as I said, geranyl transferases were used. So when some other kind of prenylation was used, um, then we observed, what did we observe? That is not given here. Okay, so these are all the studies that was done essentially. So researchers, again, they checked whether the electrophoretic mobility of the protein changed by using farnesyl transferase inhibitor. So they were trying to figure out why is farnesylation important, and by inhibiting it, if there is any change in the mobility of the protein, if there is any change in the localization of the protein, nothing was observed. So if there would have been any robust alternate prenylation machinery, let's say if we do not farnesylate the protein, but rather do some other kind of prenylation, then we might see some sort of change in electrophoretic mobility, but that was also not observed. So any kind of change. So one we saw was there was a single base substitution. Do you remember that? In the previous case, C to T change. Are you guys following this?
Yes, sir.
So in this case, the disease was caused due to a single base substitution, right? This kind of disease can also happen due to some other reasons also. We will see. So it could happen due to E145K mutation. So what is E? Which amino acid? No answers.
Can't remember it. I guess it's isoleucine.
Anybody else?
Glutamic acid.
Yeah, glutamic acid is E, and then K is lysine. So what is the difference if you see the structure? Lysine is having a very big R group, right? So if there is any change in E145K, there also we see a change in the lamin structure. So E145K patients, if we check their fibroblast, there also we see a misshapen nuclei, multiple defects in chromatin organization. So again, progeria could not be caused due to one reason alone or one kind of mutation alone. There could be different kinds of mutations that could happen. And again, in this case, dermal fibroblasts were checked. And here, the E145K there also, you can see in the controls cells, the nuclear architecture remains more or less stable, or it remains like a round form. In E145K mutation, there is a bleb structure. In E145K mutation, it affects both lamin A and lamin B both. But in case of the previous mutation, it only affected lamin A. Fine. That is what has been observed. So here also, let's see. So the clustering, the E145K mutation, it causes clustering of the centromeres and then associated methylation, but it does not alter the distribution of this methylation chromatin mark. Whereas LA50 progerin, which we observed earlier, where there is a 50 amino acid deletion, it alters both the epigenetic marks. So is that clear to everybody? Or I think I need to explain this. So what happens in this case of mutation, in this kind of mutation, E145K mutation, the centromeres, they are clustered, at the periphery. That is what is happening. The clustering, okay, sorry, the centromeres, they are usually at the periphery, but the clustering of the centromeres, you can see the dark dots, they are changed. But the methylation pattern, it does not change. In the previous example, the methylation pattern also changes, as we have already observed using Western blot. So mislocalization of heterochromatin, centromeres, and telomeres, all this mislocalization will cause an aberrant gene expression. Clear?
Yes, sir.
So this is what we observe. Then again, another thing that was observed in case of, this kind of mutation was the disorganization of the filaments. So essentially the lamins, they have this filamental structure, as you can see in the, cryo-ET, the third panel. So the wild type lamins and the, LA 50 amino acid deleted lamins, both of them, the filamentous structure at least remains the same. But in case of E145K, mutation, that lamin, the filamentous structure no longer remains. It becomes disordered. So one thing is clear, with different kind of mutations associated with a disease, we have to treat it in a different way. Okay, so in case of this kind of mutation, if we use farnesyl transferase inhibitors, it does not improve the recovery of the cells. That has been observed. So E145K mutation, what it does, it inhibits the reorganization of the interface chromosome territories in daughter cells in early G1. So that happens due to abnormal, lamina formation. So the chromosome, organization is changed in this case. So this is what we observe in this. So and this happens in early G1. What else do we have? Okay. So the abnormal association accumulation of permanently farnesylated progerin at the nuclear envelope membrane disrupts the normal functioning of the lamina. That happens, but in case of E145K patients, there is abnormal polymerization of lamins. So these are the two differences in these two mutations. So E145K mutation, there it affects both lamin A and lamin C, whereas the other kind of mutation, it only affects lamin C. E145K mutation causes lamin polymerization and assembly, resulting in a cellular phenotype that is distinctly different from progerin expressing cells. So as we have already seen in case of aging, all the kinds of repair pathways, this is non-homologous enjoining, homologous recombination, base excision repair, nucleotide excision repair, and mismatch repair. All of these, um, repair machineries, they are down regulated. That causes more accumulation of mutations, point mutations in specific. So repair of these double stranded breaks which are caused, they require these enzymatic activities of nucleases, helicases, ligases, and these kind of enzymes. So most mammals, they have REC-Q helicase, like REC-QL1, BLM, WRN. These are all the helicases which are the first step of repair mechanism. So whenever there is some problem, the DNA needs to be unwound using helicases. So REC-Q helicases, they play a very important role and they are referred to as the guardians of the genome. They maintain the genome integrity. So we are talking now about Werner's syndrome, which is nothing but adult progeria. So here there is a mutation in the WRN gene, the helicase gene, right? So it's an autosomal recessive genetic disorder. It has 34 coding exons and encodes for a nucleoprotein of around 1400 amino acids. So here, REC-Q helicase, it protects the genome stability by regulating DNA repair pathways and telomeres. So as you can see, the structure of helicases here, REC-Q, whether you have taken from E. coli or Homo sapiens, you can see at least two things are mainly maintained. One is the helicase domain, which is conserved in all the cases, and the other one is the RQC domain, important for this function. And in case of the WRN gene, here we do not have any patient samples. Okay, fine. So this is how entirely the genome organization is. So we can say that this gene is conserved across various species. So it's a very important gene. Any gene, if it is conserved across species, right from prokaryotes to eukaryotes, that means that gene is very important for the survival. So so in undamaged cells, what we see that WRN, it localizes to the nucleolus, but after DNA damage, it rapidly mobilizes to other nuclear regions where damage needs to be repaired. Then we see the acetylation of this, WRN protein by P300. It's again an acetyl histone. I mean, it's a protein acetylase. It affects its subcellular distribution, and deacetylation mediated by sirtuins, we have already studied about them, affects its translocation to the nucleolus. So helicase and exonuclease activities are affected. So Werner syndrome patients, they have, again, the symptoms of accelerated aging, including early onset of osteoporosis, arteriosclerosis, and type 2 diabetes, cataracts, all the diseases associated with aging. So we talked today about two main things. One is progeria. In case of progeria, HGPS, we saw about two types of mutations. One was the C2T mutation, which causes a 50 amino acid deletion. Then we talked about the E145K mutation. These two mutations are different kinds of mutation. Both
Sir,
causes HGPS.
can you please explain the E145K mutation once more?
What happens in E145K mutation? There is abnormal polymerization of lamins. So if you see here, the lamin structure is changed in case of E145K mutation, whereas in case of the previous one, the 50 amino acid deletion case, there the lamins, they are formed, but they form filaments also properly, but their accumulation becomes toxic because their farnesylated motifs are not, cleaved. So this is the difference.
And here it causes abnormal polymerization.
Yes.
And
Both
here it
causes
doesn't
similar
form.
symptoms, but the idea behind all of this is that even though the symptoms might be same in both the mutations, but the treatment strategies are different. So let's say if somebody has HGPS, we can't treat them right away by using some drug. So that's the idea. Clear?
So that means E145K, that like mutation of that gene, it causes abnormal polymerization of the lamin.
Right, right.
That is also a problem. And in the case of HGPS, that where C changes to T, there lamin does not form properly.
Yeah, so
Because of
mature
farnesylation
lamin
form.
is not formed, it is
Mature lamin.
isoprenylated. That's why it is toxic.
Sir, can you move to the previous slide? Wait, one once. No, the previous one. Another one. So what is this LA50 progerin? LA50 progerin, it is written.
Lamin A 50 amino acid deleted.
That's
So we talked about these things. So we talked about again HGPS, the two kinds of ways HGPS can be caused, and then we talked about Werner syndrome, that is adult progeria. So here the helicase activity is altered. So if helicase activity is altered and there is a mutation in helicase activity, all the repair, pathways, they become dysfunctional, because helicase is one of the primary, things which will come up whenever there is a DNA damage. So if helicase activity is altered, then we again see symptoms of aging coming up. Why we studied about these two, diseases in specific? Because these two diseases are they both cause accelerated aging. So if we can devise any strategies to uh, prevent these diseases from happening, then we can also, do something about aging as well. So is that clear so far? I think we have a chat here. Audio is too low. Mayuk, is there any problem?
Yes, sir. The audio is too low as I can hear, but I could see the slides. Everyone is properly visible. Just the audio was too low because I'm using my phone now due to some
Do you
network
want
issue.
me to repeat something, or would you see it from the recording? I mean, which way will be better?